All right, everyone, we'll get going. Great. Well, good afternoon, and thank you for joining us for the last biology department seminar of the fall. We almost made it. Yeah. So for those of you that don't know me, my name is Michael Joyce. I was formerly an IBS grad student, now a wildlife researcher out at NRRI. So it is my pleasure today to introduce our seminar speaker, Taylor Velander. Taylor has been a master's student at IBS for the last several years. He'll today be talking about his thesis project. We're not quite ready for defense, but we're getting very close. So he's got a lot of good stuff to share, and I hope you enjoy it today. Taylor grew up here in Duluth. He graduated from UMD with a bachelor's in biology in 2019, right? And so one of the cool things about advising Taylor, co-advising Taylor with Ron Mullen, is that certain aspects of his project we were able to start working on kind of at the end of his undergraduate career. So there's been some longevity here, and Taylor's been a really phenomenal graduate student for us. So there's a lot of qualities that Taylor has that have led to his success as a grad student. I'm just going to focus on a few of them. The first is really independence and ownership. So when we started working on this project when he was late, just about to finish his undergrad, he really took ownership of it right away. It was something that he had a lot of motivation for, and he really kind of drove a lot of this project. So we used to meet every Tuesday morning out at NRI, meet for an hour or two, kind of chat about where things were, look at some of the math that he was going through. And it was really cool that when we were done with that meeting, I knew that Taylor was going to go. He was going to follow up on things. He's going to dig deeper than I necessarily had time to. We were going to come back next week. Things were going to be pushed ahead. It was a really, really fun experience for me. He's carried that through as a master's student as well. The other thing about Taylor that really stands out is his work ethic. So really hard worker, one of those people you send him out into the field, you know he's going to do what he's supposed to do and come back having completed the task at hand. One really good example of his work ethic is for the last 7 years, he has served as part of the Duluth Air National Guard. He's currently a staff sergeant. So not only while he was an undergrad, but while he was a grad student, it wasn't just balancing teaching and coursework and research. It was doing all that while also balancing drill and deployments and things like that. And so that's really a testament to his motivation and his ability to find a way to get stuff done. And those have really made it a joy to work with him. So his thesis is really focused on understanding some of the thermal and energetic aspects of winter rest site selection of American martins, not just here in Minnesota, but across the climate gradient in the Western Great Lakes region. So please join me in welcoming Taylor.
Thank you, Michael. As Michael said, I am most interested in understanding the factors influencing martin winter rest site selection across the Western Great Lakes region. What is it? There we go. Heterotherms, which include both birds and mammals, must maintain a core body temperature within a relatively narrow range of body temperatures. Across the range of ambient temperatures that they experience, there's a relatively narrow range of ambient temperatures at which they expend very little energy in order for them to maintain their internal body temperature. This is called the thermal neutral zone. If temperatures are outside the thermal neutral zone or they're experiencing temperatures outside of it, they ultimately expend excess energy in order for them to maintain that internal body temperature. And so as you can see, there's minimal energy within the thermal neutral zone. However, it increases as ambient temperatures go beyond the thermal neutral zone. And so in order for mammals to survive, they regulate heat exchange with their environment through various morphological, physiological, and behavioral adaptations. This leads me to my study species, the American martin. The American martin occupy northern and high elevation forests within North America. They are part of the weasel family and are an omnivorous species that primarily predate on small rodents, squirrels, as well as various bird species. Why do we care about martins? Well, martins are ecologically important because they function as both a prey and a predator species with many of the ecosystems that they're a part of. They also have management value because they are used as an indicator species for many of the old growth forests within their range. They are also recreationally important because they provide some revenue in many parts of their range as well. For example, in Alaska, they generated $1 to $2 million in annual revenue because martins are a fur bearer species in that state. They are also societally and culturally important because they have high intrinsic value to the public, and they are extremely important for many of the Native American clans. Thermal regulation for the American martin can be energetically costly. This is because they have a lower critical temperature of roughly 16 degrees Celsius, which is about 60 degrees Fahrenheit. And because they live in northern regions in North America, they are often experiencing temperatures well below their lower critical temperature. They also have elongated bodies, which means they have a high surface area to volume ratio, which means that they're losing heat at a higher rate to the environment. They also lack fat stores and have relatively short fur compared to other mammal species. And so in order for them to overcome the obvious morphological and physiological limitations, they often thermoregulate behaviorally. For example, they decrease their activity levels and increase their prey size when it's cold out. They also exhibit the behavior by using different microhabitats in order to rest and reproduce during the year. And so throughout the year, they take advantage of many different microhabitat types. Above ground sites include tree branches, witches' brooms, which are essentially dense shoots that are forming on conifer trees. They also utilize nests either made from squirrel species or bird species. And finally, they also occupy cavities, which have either been excavated by other animal species or they have been formed from fungal species or through natural decay. They also take advantage of ground level sites, such as burrows within the ground, hollow logs, root masses within tip-ups, as well as log piles. Throughout most of their range, martins rely heavily on the synivium as a winter rest site. The synivium is essentially a under the snow rest site that is often associated at the snow soil interface. Deep snow conditions in most of the areas that they occupy create a synivium that is relatively stable compared to ambient temperatures and can maintain roughly 0 degrees Celsius despite changes in ambient temperature throughout the winter season. And because of this warm, stable environment, researchers believe that martins are selecting the synivium because they provide a warmer rest site than other microhabitats. Over the last few decades, however, there has been a reduction in the spatial extent, the temporal duration, the depth of snow, as well as an increase in the density of snow as a result of climate change. And so although climate change has increased the temperatures throughout the winter, the warming temperatures are likely not enough to offset the negative consequences that climate change has had on snowpack conditions. And so as snow conditions continue to change, what will martins do? Or even what will other species do? What other species who have relied on the synivium as an energy saving strategy do in response? For martins, we believe that they will make a shift in their microhabitat selection. We know that in response to seasonal changes in climate, martins choose different rest sites in their spatial position. For example, in Minnesota, martins select subnivean and subterranean sites during the fall and winter, but then switch to elevated positions during the spring and summer. Many researchers believe that this switch is correlated with decreases in snowpack and increases in ambient temperature during the switch in seasons. Even further evidence of a switch, we believe, is occurring within the Western Great Lakes region. Throughout this region, marten populations occupy a variety of forest types along a climate gradient that includes areas with deep, persistent snowpack, such as areas in the northern part of Minnesota, and then areas with low transient snowpack, such as areas in northern lower peninsula Michigan. What's interesting is that across this gradient, there are differences in rest site selection. For example, in Minnesota, the martins are primarily using classical patterns of selecting the subniveum and subterranean sites. This is similar in northern Wisconsin, where they're also using subniveum sites. However, recently there has been evidence of them using cavities as alternatives during the winter. And then finally, in northern lower peninsula Michigan, they're actually predominantly selecting cavities. Another example of this occurring is within the European pied martin species in Europe. The northern species in the colder regions, these martins are occupying subniveum sites. However, in the warmer southern regions, they're using cavities. And so having a better understanding of the causes and consequences of selecting different rest sites within the Western Great Lakes region will give us a better understanding of how martins will essentially respond to climate change. And so this leads me to my two broader objectives. Our first objective was to develop and test a thermal model that can predict the tree temperatures within tree cavities. Currently, there is a model that can predict temperatures within the subniveum. This is called Niche Map R, and it's been developed by a couple of authors, Kearney and Porter. However, currently there are no models that have yet to be made that can predict the temperature within tree cavities. And then for my second objective, I plan to use empirical data as well as these models to understand or evaluate the roles of climate, snowpack temperature, and tree cavity temperature on American martin rest site selection across Minnesota and Michigan. And so back to my first objective, developing a thermal model for tree cavities. Although we are interested in pine martins, tree cavities are also known to be an important structure used by many mammals as well as bird species for denning, resting, and reproduction. For example, tree cavities are important structures used by porcupines as well as various bird and squirrel species. Although tree cavities are important for predator avoidance, they're also an important component for thermal regulation. And so throughout the last few decades, there has been increasing interest in understanding the role of cavities on thermal regulation within birds and mammal species. And so in order for us to make direct connections between the animal response and cavity temperatures, many researchers have used data loggers to essentially measure the temperature within these tree cavities. The problem with data loggers, though, is even though they are accurate, they are limited through how long they can measure temperature for, and it's difficult to measure every single cavity within a single ecosystem. An alternative method is the use of thermal models. In general, the microclimate within any microhabitat is influenced by the fundamental processes of heat and mass transfer. That includes radiation, convection, conduction, and any displacement of air within the microhabitat. These are then in turn governed by the climate within the site, as well as the habitat features within the site and the thermal and physical characteristics of the cavity itself. And because the microhabitats all vary in these characteristics, their temperatures also vary. And so what thermal models can do is they can account for these differences in thermal characteristics and then allow researchers to predict the temperature within microhabitats under various environmental scenarios. Again, there has been models created for other microhabitats, but again, there's none for the tree cavities. And so my specific objectives for this study were first to define a tree cavity system and identify the major heat transfer processes that would allow us to develop a model that is easy to parameterize and is accurate. My second objective is to then develop a model based on the assumptions made in objective 1. And then third, test the model's ability to accurately predict the temperature within a broad range of cavities that vary in thermal and physical characteristics. So defining a tree cavity system. Currently, there are many derived energy flow equations that we can use to ultimately predict the temperature within a cavity. The problem is that a lot of these are computationally intensive. because a lot of them require partial derivation. Overall, it's just difficult to apply them. They're also relatively complex with regard to how many input parameters they require for modeling. But our model, however, we are assuming that a tree cavity is a lump capacitance reservoir of heat, which ultimately simplifies the tree cavity or the model by assuming that radiation and convection are negligible, and that there are only two main processes of heat transfer. The first is the conduction from the ambient air through the wall of the cavity and into the cavity. And then the second one is the displacement of air from the outside to the inside through the entrance hole of the cavity itself. And so now if you incorporate these two processes together, you get equation 1. I'm not going to go into extreme detail here, but overall what is happening is that if we add the two processes together, you can ultimately calculate the overall energy transferred in and out of the cavity over a period of time. And then through some derivation, I came up with a final equation. It looks pretty dense, but overall what it does is it can calculate temperature at any given point in time using ambient temperature, as well as the thermal and physical characteristics of the cavity itself as input parameters. Some of the input parameters include the specific heat capacity of the wood and the bark of the cavity, the specific heat capacity of the air, the thickness of the wood, its thermal conductivity, the surface area of the cavity, as well as the entrance hole, or even the air velocity within the cavity. And so overall, because I'm not getting into the derivation of this, if you have any questions about this equation or how I derived it, feel free to ask me after the presentation. We then validated the model by testing its performance on 43 natural cavities located within the Superior and Chippewa National Forest, as well as the Manistee National Forest in the Lower Peninsula, Michigan. One thing to note is that all cavities varied in their thermal and physical characteristics. For example, cavities were selected to either be in live or dead trees. They also varied in tree species, and they also varied in their sizes. Ultimately, having a broad range of cavities allowed us to test the generalizability of the model. We also tested the model on 27 artificial cavities that fell within 5 different design types. These were located within northern Minnesota and were part of a Fisher habitat improvement study in this region. Like the natural cavities, artificial cavities also varied in their materials that they were made out of. For example, some of them varied in the plywood type they used, and some of them contained insulation, whereas one of them did not. They also varied in the sizes of the materials they used. The plywood thicknesses also varied between these cavity types. And then finally, they also varied a little bit in their overall design. And so again, we're testing the generalizability, or basically the ability for the model to predict temperature within a broad range of artificial cavity types. Input parameters for the model were either measured or obtained through literature. Measured variables within natural cavities were done without cutting the tree down. We basically wanted to save the cavity because we're worried about limited cavities within these regions. And so we had relatively rough estimates of their physical characteristics, whereas artificial cavities, they were much finer estimates. And so you'll actually see that that's going to create a difference in how the model worked later on in this presentation. For each cavity we sampled, we also deployed 2 temperature loggers. The first was an ambient temperature logger placed right outside the cavity. And the second one was a temperature logger that predicted the temperature within the cavity. It was placed at near the bottom of the cavity and essentially was hung at the same duration as the ambient temperature logger. And then they were set to measure temperatures at 30-minute intervals. With the ambient temperatures and the input parameters we either obtained through literature or we measured, we then modeled the temperature within each cavity we sampled. We then compared modeled and measured temperatures from each cavity to ultimately assess model accuracy. Model accuracy was assessed using model error, which is basically the difference between measured temperature values and their corresponding model temperature values. We then used model error to calculate root mean squared error, which is essentially the average error across a period of time. And we did that, we calculated Rmse for the entire sampling period, as well as at the daily scale for each cavity. For analysis, we use 3 generalized estimating equation models to evaluate sources of error within the model. The first model assessed differences in daily RMSE between natural and artificial cavities. The second model assessed differences in daily RMSE between the 5 different box types. And third model assessed differences in daily RMSE between natural cavities as a function of study area, the heat capacity of the cavity, its thermal conductivity, surface area, its engine soil area, as well as its wall thickness. These covariates were selected because they represent the major thermal and physical characteristics that essentially influence the heat transfer in and out of the cavities. And the main reason we used GEE was because we wanted to test the generalizability of the model while still accounting for the correlation between sampling days. During analysis, we identified periods where measured temperatures would suddenly remain constant at 0 degrees Celsius. Here are 2 separate figures showing this occurring. If you look on the figure on the left, you have ambient temperature in the gray, model temperature in the blue, and measured temperature represented by the black line. Modeled and measured temperature appear to be very similar when temperatures are outside or above or below 0 degrees Celsius. However, as temperatures reach 0 degrees, you suddenly have measured temperatures remaining constant for a small period of time. This occurred 3 different times in this case. That's a less extreme example. A more extreme example of this occurring is on the right. Again, measured and modeled look very similar. However, as temperatures reach 0 degrees Celsius, modeled follows the daily oscillations of ambient temperature. However, measured temperature suddenly, we call it flatlining, but basically remains 0 degrees Celsius for a few days actually, and then suddenly returns back to normal. And so to see if these periods were a major source of error within our model or while modeling natural cavities, we basically identified every single day that expressed a flatlining period and then added it as a covariate within our GEE model 3. All right, results and discussion. So for natural cavities, we had over 5,000 sampling days, which equated to over 200,000 temperature measurements, and it resulted in an average of 137 sampling days per cavity. For artificial cavities, we had over 4,000 sampling days with an average of 170 days sampled per cavity. For both cavity types, we measured temperatures within a broad range of ambient temperatures. Again, testing the generalizability of the model. Overall, the model showed pretty high accuracy. Here's an example of ambient measured and model temperatures across a week in a natural cavity. For the most part, again, the model is accurate. However, we are seeing some error at the coolest and warmest parts of the day. This is pretty consistent throughout this entire week. Here's another example of a natural cavity. This one actually works a little bit better. We're not seeing that the high error at the lowest, the coolest and warmest parts of the week. The reason I think that these cavities differed in accuracy is simply because we probably had better estimates of this one's thermal and physical characteristics than the previous cavity. This is an example of an artificial cavity. Again, we're seeing the same thing. Very accurate, but there is some error occurring at the peaks and the valleys of the day. And another example where it's working even better. And again, it could be a situation where we had better estimates of the physical and thermal characteristics of this particular cavity design. When we summarize RMSE across the full sampling period, natural cavities were within roughly 2 degrees RMSE, and artificial cavities were within or around 1 degree RMSE. When we look at the daily scale, we find similar findings. Natural cavities had significantly more error than artificial cavities. We actually believe, again, and this is where the differences in how we measured them come up. We believe that the reason why natural cavities had a higher error was because, again, we had rougher estimates of a lot of their physical and thermal characteristics. And so because of this, future work could focus on getting much more precise estimates of cavities. We could cut the trees down, but again, our concerns over the availability of cavities and the fact that we still had really low RMSE means that our methods were non-destructive methods were relatively sufficient. When we compare among the 5 different box types, we find error was Error was reasonably low amongst all of them. However, we did find differences in daily Rmse between box types 5 and 1, box types 5 and 4, and then box types 3 and 4. What's interesting is box type 5's design was completely different from the rest. It contained no insulation. And so what's possibly happening here is we're not accounting for some of the heat transfer processes within this particular box. Box type 3 was also a little bit different from the rest. It contained Douglas fir as its primary structural component, whereas the other box types all contained pine plywood. And so again, what we might be seeing is we're getting poorer estimates of Douglas fir's thermal characteristics that are different from their actual true values. And so future work could focus on finding better estimates, but given that we're obtaining a lot of these values through literature or through our own rough estimates, and the model is still working relatively well, we're pretty confident in what we're finding here. For natural cavities, there was no relationship in daily Rmse amongst the covariates that we tested. Here's an example of the relationship between daily Rmse and cavity surface area. Like the rest of the covariates, we found no relationship. Again, what this tells us is that the model is extremely generalizable and basically can predict temperatures within cavities that vary in a lot of these characteristics. We did, however, find that days that exhibited a thermal arrest period contributed to significant amounts of error within our model for natural cavities, where days that did exhibit a thermal arrest period had significantly higher daily Rmse than days that did not. Given that thermal arrest periods are occurring at 0 degrees Celsius, it is likely that the phase changes of water that occur at 0 degrees Celsius is in part responsible. When we look at temperatures within the cavity reaching 0 degrees Celsius, we believe that what's happening is that the water inside of it is entering a phase change process of being between a solid and a liquid. And while it's during this phase change process, the latent heat of fusion and solidification of water is keeping the cavity at 0 degrees Celsius for an extended period of time until the water has either fully transitioned into either a solid or a liquid. Another reason we believe that the phase change processes of water is in part responsible for this phenomena is because we are seeing similar patterns occurring within the soil during winter freeze-thaw cycles. And it's also a major process that is keeping the subnivium at roughly 0 degrees Celsius throughout the entire winter as well. In conclusion, we developed a relatively simple model that is reasonably easy to parameterize. It only includes 2 main heat transfer processes. Given its assumptions that radiation and convection are negligible, the model works pretty well. It can account for the thermal and physical characteristics of the different cavities. And so overall, it's generalizable and useful. Nonetheless, obviously the model does not work very well when temperatures are around 0 degrees Celsius. And so future work could focus on incorporating phase change processes of water as part of the model. Now that we have a working cavity model, I then move on to my second objective, which is evaluating the roles of climate, snowpack, and cavity microclimate on martin winter rest site selection in Minnesota and Michigan. For the second objective, I'm going to focus on 2 study areas, the Superior National Forest and the Manistee National Forest. The reason we chose these 2 study areas is because we know that the martin populations within these regions differ in their rest site selection. For example, in Minnesota, in the Superior National Forest, they follow the classical patterns of selecting ground level sites in the subnivium or subterranean, whereas in northern lower peninsula Michigan, they are selecting cavities during the winter. And so the big question here is, why are there differences in winter rest site selection between these 2 study areas? We believe that it could be related to several factors that are not all mutually exclusive. The first, it could be related to differences in the snowpack and the ambient weather conditions between the 2 study areas. For example, in Michigan, the Manistee National Forest study area, there could be limited snow cover and relatively high ambient temperatures, whereas in Minnesota, we have relatively deep snow conditions and cold ambient temperatures. It could also be related to differences in the microhabitat temperatures. within tree cavities and synovium sites within these areas. Cavity temperatures could be warmer in the Manistee than the synovium, which is making martins want to select the cavities over synovium sites, whereas in Minnesota, it could be that the synovium is actually warmer than the cavity, which is then making the martins wanting to select the synovium over cavities. And then finally, it could also be related to the availability of the different rest sites. For example, if there is limited snow cover in Michigan, there simply may be a lack of rest sites for martins to be using in Michigan. In the study, I focus on 4 main objectives. First, I compare snowpack and ambient temperature between the 2 study areas. Second, I attempt to determine if climate is what is driving martin rest site selection within these areas. And third, I compare the temperatures between tree cavities and synovium sites within both of them. And then finally, I estimate the energetic consequences of selecting the different rest sites within these areas as well. When we compare the climate conditions over the last decade, we find that the Minnesota study area, indicated by the darker bars, is much colder than the Michigan study area. It also has relatively more deeper snowpack. It maintains deeper snowpack throughout the winter. However, it appears that Michigan still maintains relatively high snow depth throughout the winter. For example, if you look at January and February, it's roughly 37 centimeters deep, which is about 16 inches. Based on a study done in Wyoming in 1994, 2 authors, Sandra Taylor and Steve Buskirk, predicted that martins should be selecting synovium sites when the snow depth exceeds 15 centimeters and the temperatures fall below -5. If these conditions are not being shown on a particular day that they're resting, then they should be selecting a tree cavity. One thing to note is that these predictions that they made were based on the assumption that synovium rest sites are warmer under these particular conditions. If these particular conditions were not being shown, then tree cavities would be warmer than synovium sites. Given what we know about differences in the snowpack between these 2 study areas, it is likely that these differences in climate conditions could be leading to the differences in rest site selection between these 2 martin populations. And so to determine if martins are truly selecting these microhabitats based on snowpack and ambient weather conditions, we compared predictions from their model to that of actual microsite selections made by martins in these 2 study areas. To do this, we gathered data of resting events that occurred within each study area. These were gathered from telemetry data that occurred in 2 different studies. One of them was my advisor, Michael Joyce's master's project. And then we compared the model predictions made by their model to the actual resting events. And so ultimately, if martins are selecting a site that the model predicts, what this tells us is that martins are largely selecting sites based on the differences in snowpack conditions and ambient weather. In Michigan, we found that martins are largely selecting microhabitats based on the snow depth and ambient temperatures, where 84% of the time, the actual rest sites they used were predicted by Taylor and Buskirk's model. However, there were instances where Taylor and Buskirk's model predicted that they should be selecting the synovium. However, martins were selecting cavities. And then there were also situations where the model predicted that they should be using a cavity, but martins were actually selecting the synovium. In Minnesota, the results are a little bit different. We found that martins in Minnesota were selecting sites based on snow depth and ambient weather only about half of the time. The other half of the time, there were conditions where snowpack and ambient temperatures would have forced them to be using a cavity. However, they were still selecting a synovium site. What is interesting to point out, though, is that a lot of these resting events, Michael found that they were actually using burrows as well. And so what could be happening is that Taylor and Buskirk's model might not be accounting for the soil on top of the burrow as an important input parameter for how they're selecting their rest site. And so in reality, martins could have been selecting the warmest rest site, but the model just could not predict it. Overall, however, there were conditions that we found where snowpack and ambient temperatures should have forced them to select particular sites. And again, they were not selecting sites based on the model predictions. And so what this could mean is that there may be limitations in Taylor and Buskirk's model, or it could be that we need to actually take a closer look at the microhabitat temperatures within these two areas. Which leads me to my next objective. In order for me to test the differences in microhabitat temperature, I basically measured the temperature within tree cavities and subnivium sites throughout both study areas. And then I assessed the differences in daily temperature within these sites using one-way ANOVA. What we found is that in both study areas, the subnivium is on average the warmest microhabitat. So in Minnesota, quite a bit warmer than cavities. In Michigan, quite a bit warmer than cavities as well. When we compare between study areas, we find that cavities in Michigan are quite a bit warmer than cavities in Minnesota. This could be related to differences in the insulation of the cavities between the two sites. However, I performed a t-test comparing major characteristics between the cavities. We found no significant differences in their characteristics. And so what this basically tells me is that the reason why cavities are warmer in Michigan are likely simply because the ambient temperatures there are warmer. Same thing with subnivium. The subnivium site is quite a bit warmer than Minnesota sites. And so again, it could be because of ambient temperatures. And so what does this all mean energetically? To do this, Michael actually developed an energetic model to calculate a Martin's energetic loss during inactivity. Essentially, the Martin's energetic loss is the sum of the Martin's basal metabolism while at rest, plus any other thermoregulatory losses that occur when temperatures are below their lower critical temperature. And again, like the cavity model, I'm not going to go into detail on how this was derived, but one big thing to note is that a lot of these values here were obtained through laboratory studies on Martin metabolism. Using this energy loss equation, I estimated the average energetic loss of a female and a male martin within each microhabitat type and across both study areas. While applying it, I assumed that martins are inactive for 19 hours a day and that their only period of activity was between 10 a.m. and 3 p.m., which basically was assuming that martins are only active during the warmest parts of the day. Like the microhabitat temperatures suggested, when the subnivium is available, martins should be saving more energy if they were to select the subnivium over cavities. In Minnesota, they would be saving about 100 kilojoules per day if they were to select the subnivium over a cavity, which is equivalent to about 1 redback bull, which is a major prey species that martins select in these areas. In Michigan, martins would actually be saving only about 60 kilojoules per day if they were to select the subnivium, which is about a half of a redback bull. And so this information, coupled with the fact that the cavities in Michigan have the same energetic cost of, if a martin were to select a cavity in Michigan, it would have the same energetic cost of a martin selecting a subnivium site in Minnesota. And so what does this tell us? Well, if snowpack continues to deteriorate in Minnesota and temperatures warm in Minnesota, it is likely if a martin was forced to select a cavity in Minnesota, they would not experience a huge energetic loss. Overall, though, there is an energetic reason for why martins should be selecting subnivium sites within both study areas. But again, we're seeing that martins are selecting cavities in Michigan. And so why is that? Well, we believe that it's an availability issue. We know that the formation of subnivium sites are often associated with coarse woody debris. Coarse woody debris is downfall, basically down trees or branches that are littered across the forest floor. And what happens is it allows the creation of air pockets within the subnivium, and these are essentially rest sites that martins can use when they are available. In Minnesota, there is an abundance of coarse woody debris in the Superior National Forest, I should say. Even in that area, there's all the soil allows for the use of burrows. In Michigan, however, until the last few years, before the last few years, there has been a lack of coarse-weighted debris. And there's all the soil there is relatively sandy, which means that burrows, it's difficult for burrows to be created. And so there's a lack of burrow sites for them to use. And so overall, what does this mean? Well, we know that snowpack is present in Michigan. And so if there is a lack of coarse-weighted debris and burrows, there just may be a lack of synovium sites for them to be using. And so they're being forced to select cavities due to the lack of availability of synovium sites. Consequently, there is future work that is needed. The data that I presented today was all empirical data taken from a single year, this last year. And so future work would focus on using the predictive models, the synovium model, as well as the tree cavity model I developed to predict temperatures within microhabitats within both of these regions during years that exhibited high snowfall or differences in snowfall and differences in ambient temperature. And then because there is preliminary results suggesting that there is an availability problem within Michigan, future work would also focus on measuring the availability of these different rest site types in both of these study areas. With that, I'd like to thank my advisors and committee members, Dr. Ron Moen, Dr. Michael Joyce, and Dr. Guy Sander. I'd also like to acknowledge all my financial and logistical support from the Integrated Biosciences Program at the University of Minnesota Duluth and the Natural Resources Research Institute. I'd also like to thank my funding sources in the Minnesota Environmental and Natural Resources Trust Fund and the Great Lakes Restoration Initiative administered by the Bureau of Indian Affairs. And finally, I'd like to thank my collaborators from Michigan, Paul Keenlands from the Grand Valley State University, and Bob Sanders and Angela Kujawa from the Little River Band of Ottawa Indians. Thank you. Do you have any questions?
I'll leave the light off for now. We can turn it on as needed, but good job, Taylor. So in the tradition of the biology department seminar, we'll take our first questions from undergrads. This is one of those tricks I remember from TAing where you just have to be quiet long enough until people feel uncomfortable. Undergrads. Now it's getting where I'm uncomfortable. We'll move on to grad students.
So one question that I had was, when you were measuring the temperatures within the cavities, did you ever either have the ability or the chance to actually look at if something actually inhabited that cavity to basically change the thermodynamics of that cavity?
So tree cavities, yes, there were many instances, and I didn't bring this up because for sake of time, but there were many instances where for the cavity developing model, the cavity model part of my thesis, we tried our best to eliminate animals from entering, but there were instances where suddenly you'd see measured temperature shoot up to ridiculous values, like 98 degrees, like Fahrenheit, which is probably the body temperature of a lot of animals, right? And so a lot of those periods were eliminated from analysis because there's no way it didn't look right compared to the rest of the measure.
Just to add on, we're also in Michigan, they're actively tracking martins, so they knew that some of their study areas were in some of the cavities.
And we did, even our collaborators told me instances where, because they're tracking martins, they actually had martins using some of the sites that we were measuring at the same time. So I do have some data that shows that when a martin is present, the temperature shoots up quite a bit. And so that kind of confirms that an animal is using the site. Often what's interesting though is in Michigan, we saw that occurring more there than in Minnesota. And so again, because martins are relying a lot on cavities, we're seeing a lot of that happening there, whereas because martins are using synovium here, we didn't see that much.
Is there any difference in predation views that there are prey species too? Is there any difference in predation pressure between sediment and using burrows like that versus using
That's a good question. I think Michael would have a better idea of that. I would, in my personal opinion, well, I know for fissures, for example, the opening of the hole in the tree cavities, it plays a big factor in whether they're going to be predated or not. If the hole is small enough, martins don't have to worry about fissures attacking them, or maybe even some bird species such as owls. However, I'm going to guess in the synovium, because it's less structurally supportive, I have a feeling that foxes and fishers and bobcats even probably have a higher chance of killing a martin when they're on the ground. Does that sound right?
Yeah, I can answer more later. I don't want to steal his show.
I mean, he knows more than I do about that. Any others?
That
That was your question? Yeah.
Wonderful presentation, Taylor.
Thank you.
So your ambient temperature loggers on the outside of dens, is there any way for them to track the thermal conductivity of the air, whether it's due to humidity or
These particular temperature loggers only measured ambient temperature ambient temperature. However, there are ambient temperature loggers that allow you to measure humidity within the air.
does the thermal conductivity of the air change enough to be significant to the accuracy of the model?
Well, thermal conductivity the way I see it is more from solid surfaces. Usually what's happening, you can have some thermal conductivity occurring within the air, but it's not
Like
as much as through a solid
convective
surface.
heat loss from the air increased with humidity or
I haven't even looked into that because it's not part of this model, but I'm sure it does vary based on moisture. I know, like, for example, the thermal conductivity of wood, if it's more moist, it differs than when it's dry. And so I'm going to guess that humidity does play a factor. And so this model doesn't incorporate it because we think it's a negligible thing to include, basically.
One other add-on, so the specific heat of air changes with temperature and moisture and pressure. One of the things Taylor didn't mention because of the complexity and trying to have something tractable here is within the range of pressures and temperatures we're looking, there's not a big difference in specific heat of air.
Yeah.
So that is something with
Yeah.
moisture content and all everything.
That and air density. It varies very little between -30 and 30 degrees Celsius. And so we used a constant value, basically. Is there a question? No? Oh, somebody, yeah, Aaron.
Hi. So I imagine how accessible are significant cavities? And then in terms of practically measuring them, I know that you weren't just measuring significant, but measuring a cavity versus measuring a significant, how the methods differ, and how often they just go after this view?
Well, we actually didn't, because it's difficult to actually find a true synovium air pocket within the snow. A lot of times they are created through other mammal species, such as small rodents who use the synovium a lot, or even air pockets from CWD. What we did is we used PVC pipes, and we basically mimicked an air pocket within the snow. And then at the end of the PVC pipes, we basically put mesh just to prevent animals going in there, but it allowed airflow to happen in there. And so it allowed us to mimic that of a synovium site.
We'll still keep asking grad students, but after this, we'll open up to everyone.
One question, so for something completely different now, I noticed, so for this study, obviously we're looking at the effect of winter, but would any of this also be able to translate to habitat use and cavity nesting in summer? Do they cavity nest in summer?
Oh yeah,
Okay.
yeah. So again, during the winter, they primarily, in areas with deep snowpack, they primarily use synovium sites, but areas where there's a little bit less snowpack or just it's transient, it's not as persistent, like Michigan, they're using cavities, but during the spring and summer, they often use cavities as rest sites as well as reproductive dens. And so they use cavities throughout the entire year. They just primarily use them more during the spring and summer. Yeah. So another reason for why we think martins are selecting cavities in Michigan is because of the lack of snow.
I'm curious for your model that you made, how did you exclude certain factors, like for instance, I'm thinking of entrance hole orientation?
Okay, well, and so again, I'm no engineer, but I know engineers make a lot of simplifying assumptions. And so you can, am I wrong, Dr. Sanders? I'm not wrong. And so you can possibly incorporate the orientation of the hull, but a lot of that likely has to do with, you're worried more about convection and radiation when you do that. And because this model is a lump capacitance reservoir of heat, we're assuming that radiation and convection are negligible. However, again, you can modify that. A part of the equation accounts for the air displacement from the outside and the inside. And so if you have, for example, a entrance hole that's more westward facing, where you have a lot of high winds from the west, you can modify the speed at which air is entering the hole through one of the parameters of the model. Mm-hmm. Yep.
Any other questions?
So you mentioned that the thermal arrest periods are kind of times where the model is less accurate or less,
Yep. Mm-hmm.
yeah, less accurate.
It's not necessarily less accurate because it all depends on ambient temperature. Because if ambient temperature is also flatlining, model temperature is also flatlining because it's a function of ambient temperature. And so we are seeing higher error at, yep.
Around those temperatures that have a phase change of water. So do you, and Ben was earlier asking about relative humidity, which would kind of dictate how much water vapor is within the air in the cavity. So do you have any intention of like incorporating that in the model in the future? Or?
Yes, well, at least trying to identify if humidity is a role.
Yeah.
With this, and this is actually occurring in the synovium. It's not necessarily humidity. It's more of it could be the moisture within the cavity walls themselves. And so one big thing that I didn't mention, I was hoping somebody was going to ask me, why didn't I see this in artificial cavities? Well, artificial cavities are far more dry than natural cavities because they contain plywood that is dry, and the wood chips in there are dry. And so the reason why we're not seeing flatlining artificial cavities is simply because we think it's just overall drier in there, whereas the wood of natural cavities are green, right? They have xylem, phloem, a lot of nutrients, and basically aqueous solutions going through there. And so the water is likely in the walls are likely contributing to the flatlining or the thermal rest.
Is that reflected in cavities in dead trees versus flat?
And this is difficult because a lot of studies say that snags or dead trees are drier. However, we did see this occurring in some of our snags, but our sample size wasn't huge for snags. And so again, future work could focus on seeing how moisture differs between snags and live trees, and how much moisture varies even within snags that have been dead for 20 years versus dead that have been dead for 10 years, right?
Did the times when you had the thermal arrest, was it, did it occur more during certain months? Or?
It was actually, well, a lot of it was occurring, well, it's only when the reason why, because ambient temperatures need to be around 0 degrees Celsius in order for the cavity itself to be around 0 degrees Celsius. And so in Minnesota, because our temperatures are usually negative, we actually didn't see a lot of thermal arrest periods occurring in Minnesota sites. However, Michigan sites, this last year, the time period that we measured these in, when we were looking at ambient temperature was roughly 0 degrees throughout the entire sampling period. And so there was no particular month that it was showing it. It was actually more based on whether the temperatures were around 0 or going from negative to positive, just going back and forth. Because that's essentially what creates the thermal arrest period. And that's a thing often seen in the freeze thaw cycles within soils. Yeah.
One more, otherwise we can call it there. Anyone have a burning question? All right, thanks again.